Good evening, everyone, and welcome to Pint of Science 2021. It's great to have you all with us here. We are signing in from Cambridge this evening, and we're the Our Body team. I am Anna. I'm a PhD student here at Cambridge, and I'm delighted to be your host this evening. We have two wonderful speakers set up for you tonight, Dr. Rob Finn and Dr. Pippa Corrie. And they're going to be speaking to you about the microbiome, or the world within us. And they have two really great talks lined up for you. But before we get going on that, a little bit about Pint of Science. So usually, if any of you know from before, we would have all been in the pub together this evening at a location somewhere in Cambridge. Unfortunately, that isn't the case this year. And although it's a shame that we're not all together having a pint in the pub, the beauty of this does mean that people can sign in from all over the country and all over the world as well. So it would be really great if everyone can comment in the YouTube comments next to you where you're signing in from this evening. And if you like, you can also pop in there what drink you would be having if you were in the pub tonight and what you may have with you now. So let us know where you're signing in from. In the meantime, before we get going with our speakers for the evening, in true pub fashion, we have a bit of a pub quiz on the microbiome. So for those of you who aren't too familiar with what the microbiome is, the definition is all of the microbes, so the bacteria, the fungi, etc., that live inside or on your body. So with that in mind, we have a little bit of a pub quiz to get you going. And a few questions that we would love you to start answering in the chat as well. So if we have the questions up as well. And the first question we have for you this evening is how much of our body weight is down to the microbiome? So the answers, or the answer options, are 500 grams, 1 kilogram, or 2 kilograms. Now, considering that this is just the bacteria and the fungi and whatnot on your body, it's a little bit terrifying that any of those weights are the correct ones for how much we have in the body. The second question we have, we've already got some answers coming in for the first one there, some pretty high guesses as well with the 2 kilograms coming in. But the second question we have, and keep in mind that the weight is quite high, is how many microbes are there in the body? So again, just to remind you, these are the bacteria, the fungi, all of the things we usually think we want to keep away from the body. Would you say that it's A, 5 million, B, 100 million, or option C is 100 trillion? Now, a bit of a hint for this one, as all of these numbers are a little bit out of my understanding, a bit of a hint is that we have 15 million, approximately 15 million human cells in the average body. So on top of that 15 trillion, how many microbes do you think we have as well? And I can see lots of comments coming in here. One from Jill saying 100 trillion, going straight for the highest answer there. And the third question, so this is a bit more of a philosophical one, and this is one that your answer may change throughout the evening. So based on Rob and Pippa's talks that you'll have this evening, the question we want you to think about is, should we consider the microbiome as a part of you? So is the microbiome a part of the human body? And we'll also be asking our speakers this evening what they think. We've just had a comment here from Kirsten saying yes, that's a definite yes from the get-go, and I can see lots of other comments of people saying yes. So perhaps you all know a little bit more about the microbiome than some of the rest of us. But this is something we'll be coming back to at the end of the evening, and some of our speakers will be revealing these answers as well. So without further ado, we will introduce our first speaker for the evening, Dr. Rob Finn. And it's my pleasure to introduce Rob this evening. Dr. Finn leads the Microbiome Informatics team at the European Bioinformatics Institute as a part of the European Molecular Biology Laboratory. He obtained his PhD in biochemistry from Imperial College London, and his group is responsible for several really amazing informatics resources, including Magnify, which focuses on metaomics, and Hammer, which is a fast protein sequence similarity search tool. Rob has a wildly impressive publication record with over 130 papers and almost 50,000 citations, including papers published in Nature, Cell, and Science. And interestingly, he also has a research group that probes the new functions of the microbiome, or the so-called microbial dark matter. So I'll now hand over to Dr. Finn for his talk, which is entitled The Human Microbiome, How We Study the Microbes That Have Made You Their Home. So over to you, Rob.
Okay, Thanks very much, Anna. I take it you can see my slides and can hear me.
Yes.
Perfect. Okay, so thank you very much for giving me the opportunity to speak to you tonight. And what I'm going to talk to you very much about is how we study these microbes that have made you their home. Now, if you haven't picked up on it, I work at what's called the European Bioinformatics Institute, or put it another way, I'm the computer geek who was also studied biology, and I fuse those two disciplines together. So the human microbiome. So we've already had some really sort of nice introductions from Anna. So we are talking about all of those microbes. Now, when I talk about microbes and bacteria to people, you may be thinking of something along this slide, where we have quite often, you'll see if you're going to study bacteria, you plate it out and you streak it out to produce these individual white blobs on this red agar plate. And that represents a single colony of that bacteria. And this is quite a typical sort of, slide that someone might, think about when you're actually talking about a bacteria. But actually, when we're actually thinking about this, I'm thinking about the microbiome, the whole set, and why is it so important? So it's estimated there are 10 to 100 trillion cells in your microbiome, and that means that there are at least a 1-to-1 ratio of numbers of cells in the human body compared to the microbes. But really what's always fascinated me is the following. So the human genome encodes about 22,000 genes, and whilst you and I may be about 99.9% identical genetically, our microbiomes, depending on our lifestyle and if we've had a course of antibiotics and other things, may differ up to 80 to 90%. And those microbes have a huge repertoire of genes that they encode, and it's estimated that it's over 50 million different genes that they encode, and there are thousands of different species that are found in the human body. It's also been implicated, the human gut microbiome has been implicated in huge numbers of diseases, and the numbers are constantly changing, and the different diseases where there is a causal relationship, and that can be up to 90% of different diseases associated with the health of the microbiome. But it's also very important to remember, not only is your gut microbiome implicated in disease, it's absolutely pivotal for food and nutrition and digestion and maintaining a healthy state. So as I say, there are many correlations between the composition of your gut microbiome and disease. So diseases like inflammatory bowel disease, obesity, diabetes, there's a natural close approximation between the gut microbiome and those diseases. So inflammatory bowel disease, you're in the bowel, that's where your microbiome. But it's been linked to other cases like cardiovascular diseases. And there's also this notion of the gut-brain axis. This is very sort of early phases of research. And I think there are a lot of correlations rather than actually direct cause and effect. And so there is this notion of the gut microbiome being influenced by influencing Alzheimer's, Parkinson's disease, depression. But we also have to remember that usually when you have these diseases, you can often be receiving drugs which can change the composition, and we run into this slight cause and effect. So what I want to talk to you today about is how we are now studying this gut microbiome. But the role of the gut microbiome in disease is not new. So since medieval times, it's often been the fact that if you had chronic stomach cramps, that actually monks would often tell you to go and take a good course of honey. And the reason for that is the fact that there are antimicrobial properties of honey. And actually what was causing those stomach ulcers was this bacteria, Helicobacter pylori. It caused stomach ulcers. And so it was, the idea of this gut microbiome influencing your health is not a new thing. However, with modern technology, we want to start understanding all of the microbes that are in the gut. So there are about 100 billion bacteria per gram of wet stool. So how do we actually start analyzing those? So the traditional way of actually analyzing this is like I showed you on my first slide, is where you'd actually take a stool sample, you'll streak it out onto one of those plates, you get an individual colony, and then you'd culture it. And then once you've got that in culture, you would apply modern DNA sequencing technologies to get access to the genomes of that microbe. However, culturing can be really, really difficult because you need to understand what bacteria likes to eat. And so that really limits our access, and it can be quite a laborious process. So there are two other approaches, and these are the approaches that I've been interested in. So one approach is metabarcoding, where we can get a fingerprint. Now, it's always been difficult to explain this slide, but actually with the current ongoing pandemic, most people have heard of a PCR test now. And actually that's where you're actually just amplifying a bit of the coronavirus. And that's exactly what we do here, but for bacteria. Or we can do what we refer to as metagenomics, where you actually sequence everything. So again, going back to the pandemic, you've probably heard the government referring to whole genome sequencing, in the UK having fantastic capacity at whole genome sequencing. That's what we're doing here, but we do whole genome sequencing of the lot. So these are the ways that we like to tackle it. And what I'm very much interested in is this very expensive approach, the metagenomics approach, because it doesn't rely on culturing, and we can try and get to the access of the genomes and actually understand who's there and what they're doing, rather than a fingerprint where you just get an idea of who was there. You don't get an idea of what they were doing. So how much of the gut microbiome do we currently know? So a couple of years ago, a number of papers came out in close succession where they were describing large increases in the numbers of different cultured bacteria from the gut. And we were seeing numbers of 1,000 different genomes coming out. And so one of my close collaborators, Trevor Lawley, he and I, so Trevor Lawley has been very much using culturing techniques to actually get access to these microbes, which is absolutely essential for mechanistic and actually testing these. But we were discussing one day and we said, well, how many really do we think there are in the gut microbiome? And this is where we started to use computational techniques to try and access this. So what do I mean by this metagenomics, and how can we get to genomes from there? So on this slide is a schematic of what we do. So what comes off modern DNA sequencers is these short fragments. And then what we try and do is use those short fragments and look for overlaps to assemble those into longer pieces of DNA. And then we try and group those longer pieces of DNA into sets that have come from the same actual microbe. And this process is called binning. And then we have these potential genomes, or things that we refer to as metagenome assembled genomes, or MAGs. If that doesn't really mean anything to you, think of it this way. I've essentially got a set of jigsaw puzzles for each one of the microbes. We empty out all of the pieces. We throw some of those pieces away. You take away the images of those microbes, and then computationally, I have to piece all of those jigsaw pieces back together again and get to the actual picture of the microbes without that actual information of what I'm trying to do. So in my team, we did this and looking at a lot of different samples. So we started off with over 13,000 metagenomes. That's about 1 trillion DNA fragments we pieced together. And ultimately, this binning process yielded over 240,000 genomes. But then how do we know that we've actually got that right? Or how, have I just managed to muddle up all the jigsaw pieces? So one of the ways that we do this is to actually filter. So we try and focus on the very best ones, the things that we think are complete. So for example, if we go back to the jigsaw piece, have we got all of the outer edges? We start noticing then that we think we've got something that's complete. And the other thing that we do is we can compare to sets of isolate genomes. And this is where Trevor Lawley, again, had a large culture collection, and we could do this almost blind test against these isolate genomes compared to these computationally derived genomes. So what we did is we did this at scale, and what we had is we had nearly 40,000 of these high quality mags, and we compared these to the culture collections. And what we could find is there were lots and lots of good matches to these things which had already been cultured and sequenced in isolation. And on the left-hand side, you can see a phylogenetic tree of all of the bacteria. Around the outer ring in different colors of the class or the taxonomy of those bacteria. And then the whiskers that come off that plot show the numbers of independent times that we'd actually seen that same genome. So what you can see here is we got a good match with these genomes, and we could see this over and over again. So we were really pleased that actually we could do this. We also identified that there was nearly 12,000 that were unclassified. And so then the question is what are those? We got 30% that represented something that looked like they could be new. Cutting a very long story short, we identified nearly 2,000 novel gut bacteria. And that's what's shown in this slide. What you have here is a phylogenetic tree in the middle. It looks slightly different to the previous one because we have to use slightly different techniques. But what this shows you is a mixture of isolate genomes, which is this blue color coming off the tips of the tree. And then you've got two other colors. There's a green color that shows that they are near complete, and red where the quality is not quite complete, but it's still not a bad representation. What we have is around the outside is the different phylum is indicated by the color, and then we have whiskers here, and they are a little bit less common than on the previous slide. So we found that there was a huge amount of diversity in the firmicutes. So a lot of these are spore formers and sit in the gut. We also noticed that these mags, these metagenomes, metagenomes assembled genomes are a little bit at low abundance. But one of the other things is we found first representatives in the gut for 4 different phyla. These are not novel phyla, but they hadn't been observed in the gut before. So in total, from this work, we were at 2,500 species that we could identify in the gut. So we had 553 that had been cultured, and then we've nearly quadrupled that with the number of these uncultured species. So one of the questions is why had we not seen these before? And so what we can do, because we have the genomes, we can look at the functions of these. And here's just one example of a phylum, the Actinobacteria. We could ask, what's the functional difference? And so in this plot, we show different high-level functions. And if you are in the positive direction, so you're going to the right on this slide, you're more common in a metagenome assembled genome. And if you're going to the left, that function is less common. And what you see in bold are 3 different functional groups, high-level functional groups, as indicated by these green arrows, that are actually all to deal with all dealing with oxygen and that exposure of oxygen. And we believe that actually these mags have a lower tolerance to oxygen, and that's another reason why they have not been cultured in this process through the culturing techniques, and that's why we've been able to get to these genomes. So we've not been the only group to do this. There's lots of scientists out there doing this. There were 2 other studies that came out at the same time as ours. And what we have been able to do is amalgamate all of these. And that gave us nearly 300,000 genomes. We can cluster those into representative species. And so we've now got just over 4,600 species in the gut microbiome. And that encodes a total of over 170 million unique genes. So if we go back to that original figure that I showed you, this estimate of thousands of bacteria is about right, but the number of genes was probably a little bit low. So just finishing up on the gut microbiome, one thing is that although we've got a lot of these novel genomes, what we know is extremely biased, and this is indicated by this this map, where the red colors indicate where there's loads of different genomes. And so North America, Europe, and Asia, but specifically China, we have good sampling. But you'll notice that continents like Africa have very, very few samples. And one of the things we do know is that geographically, you get very different gut microbiomes in geographical different areas, and that is to do with environmental influences. So moving now away from the gut, we also see that actually skin microbiome is another area where there are lots of interesting organisms, but there it's a very different microbial community, and you don't just get bacteria, you get far more eukaryotes, or these fungi growing. And so we've applied a similar approach to recovering the genomes. And what I would say about the skin is there's not so much novel diversity, and I think that's partly because of the accessibility of the skin, and also it's exposed to oxygen far more, and so there has been far more culturing there, and therefore we can still get access to hundreds of new mags in the bacterial area. But where we've been pushing the area for the skin microbiome is actually looking for eukaryotic organisms. And the skin microbiome is much harder to study than the gut microbiome, because it's much lower biomass. You don't have anywhere near the number of bacteria living on your skin than you do on the gut microbiome. So what we've been doing is trying to access these eukaryotes, and both Sarah and Paul have been working in different areas. So Sarah is a medical PhD student, and she's been working at actually assembling these genomes, and Paul has been developing the tooling to allow us to access these genomes. And they're much harder because the eukaryotes are bigger and they have more repetitive regions, and that's very hard to access computationally. And the microbiome is very much about, viruses as well. And now everyone's far more aware of viruses, and so we also have huge numbers of viruses in our gut and in our skin. So we've identified over 142,000 of these phages. Now these phages are really interesting in the fact that these actually go into bacteria, and they can change bacteria. They can insert their genomes within the bacterial genome, and they can also can just reside in the cell. But both of the different lifestyles can really change the composition. There's also some jumbo viruses in these microbiomes, and they have really interesting aspects for medical purposes, because you can actually load genes onto those viruses. So where's all this research heading? So there are lots of different things that influence your microbiome, whether you take medicines, the food you eat. You can also try and manipulate your microbiome yourself through abiotic things like drugs, food, prebiotics that you may consume, or even biotics. So you may buy probiotics and have live yogurts where you try and take that. But there's an increasing thought about, can we identify species that can be very helpful, sort of nurse a species, or use these phage for manipulation? But one of the things that we do know is that, you know, some of these disease states, you can take a healthy donor and you can do a fecal microbiome transplant and actually recover a disease. But as I've mentioned, that phages and bacteria, they can carry things around in their genomes that you may not want. You may have antibiotic resistance. And so actually just doing a whole fecal microbiome transfer is quite dangerous because you don't know what you're transferring. We're still discovering the entire composition. And so there are efforts to try and identify defined key bacteria that you can then put into a pill to restore that disease state. And finally, our microbiomes go far beyond. And so one of the other areas we're working on is actually looking at other animal systems for food production and how that microbiome can actually improve our food system. So I'm involved with another project, so Hollow Food, where we're looking at how we can actually improve animal production through the manipulation of the gut microbiome. And that just leads me to thank everyone, particularly Alex Almeida, who conducted a lot of the human gut microbiome, all of my team that are on the left-hand side, Bryony Jackson, part of the public engagement. And then with All Science, it involves many different collaborations and funders that are shown at the bottom of this slide. And thank you for listening.
Thank you so much, Rob. That was really fascinating and quite amazing to see how much you can do informatically and with all of these new technologies now. It's amazing anybody ever managed to do anything at such a high scale before all of this was available. So really, really amazing.
Thank you.
We have lots of questions from the audience, so we'll get going straight away with those, I think. So the first one has just popped up here, is from Rachel Turpin, and in good relation with the pub fashion here, with everybody probably with a glass of something. Her question is, what does alcohol do to your microbiome?
Yeah, that's a good question. I mean, there are lots of positives and negatives in alcohol. So it, you know, with a typical, so I'm thinking maybe a pint of beer, there, you know, the alcohol content's not massively high, so it's probably not going to do a drastic change to your gut microbiome, but you're certainly going to fuel your microbiome very strongly with all the goodness that contains in a pint. I think with all things, if you take it to an excess, you will start shifting and perturbing your microbiome in certain directions, in terms of, you might find that actually not all of the bacteria enjoy excessive amounts of alcohol in the system, and that actually you start reducing the diversity, and that diversity is quite important for health.
Great. So that's a bit of a go ahead from a scientist that we're okay to be hitting the pubs now the lockdown's reduced, but in moderation.
Exactly.
Great. And we have some more questions coming in. There's one here from Kerry Howarth, and she has said, what makes a microbiome healthy?
So I would say that we don't know exactly that answer. We know trends that make it healthy. And this is one of the things that we really want to get to. And this is where we're building up these large collections to understand. One of the key things is having a lot of diversity of that. So you have a range of different organisms. The other thing that makes it healthy is having a range of, you know, a balanced diet, lots of goodness that keeps those microbes nicely ticking over. And also trying to avoid the use of things like antibiotics needlessly, where that can really affect your microbiome. You can take out a lot of those, a lot of your microbiome, or suppress it, and then, you know, it will take a time to recover. So really it's having a good range of different microbes such that they can, you know, protect and, you know, harness all that food for you.
Yeah, that's so interesting. We think of antibiotics as this mega reliever of all of the issues we have. So it's important, I think, to consider the effect that it has on the microbiome, especially now we're understanding a bit more how important it is for you. That's really interesting. And I think we have time for one more question. So we have one here from Alex Finney. So he said, was interested to know how many of the known and, sorry, let me start that again. Was interested to know how many of the known, how many species you guess would be human specific, i.e. never found in any other animals or biomes? That's a really interesting question.
Yeah, so what we find is that the vast majority tend to be human specific in terms of, but what we see is when we do the comparisons, it may not be, you know, the same species, but they belong to the same genera. So the next level up in our taxonomic classification. So, you know, so it's really that they're very closely related. And what you do find is that actually some of these are, you know, there are general trends, but there are specific microbes that are specific to humans, and there is a little bit of most likely host adaptation going on that allows them to particularly, you know, go into that niche and occupy it.
Right, that's so interesting. So they're thriving sort of particularly in one organism, and maybe doing a bit better there.
Yep.
That's great. Well, thank you all so much for the questions, and we will actually be coming back to some more. So at the end of the evening, once we've had both of our talks, we'll be inviting Rob back on to answer some more questions alongside Pippa. So for now, just to say a huge thank you, Rob. Fantastic talk and some really great questions there. So we'll see you a bit later on.
Thanks very much. Thank you.
Great. So we're going to take a couple of minutes now just to have a look where people are signing in from. So we had loads of great comments at the beginning about where people are listening in from today. So we have Sarah from Chamonix in France. That's not a trip you'd maybe have been able to do if we were in the pub. We've got Pete saying hello from Leeds, and he has a pint in his hand and was ready for the talk. So that's brilliant. Hi from God's own country, Yorkshire. Hi to Andrew. It's good to have you with us. Signing in from Guildford, Surrey, and drinking red wine. There we go. Lots of benefits of red wine for the microbiome, I think. So that's an apt one. And then we have Lynn from Bedford with a G&T in hand. A good diversity of drinks here, in line with the diverse microbiomes we're all trying to achieve now after Rob's talk. Fantastic. So it's amazing to see we have people signing in literally from all over the world. And I think we have almost 120 people at the moment. So way, way more than we'd been able to reach in Cambridge, which I think is a real bonus of being online this year. So we'll move on to our next speaker, and we have Dr. Pippa Corrie joining us. So Dr. Pippa Corrie is a consultant medical oncologist here at Addenbrooke's Hospital. She's also an associate lecturer at the University of Cambridge. She started her medical training at St. Thomas Hospital in London, where she undertook a PhD in anti-cancer drug development and subsequently qualified for medicine at the University of Oxford. She specializes in melanoma and pancreaticobiliary cancers, and holds roles in the National Institute of Health Research and the National Cancer Research Institute, as well as being the chief investigator of multiple academic and industry-sponsored national and international clinical trials, one of which is the MITRE trial, which is aiming to evaluate the microbiome and using it as a biomarker of potential success in cancer patients receiving immunotherapies. So without further ado, I'll pass over to Dr. Pippa Corrie for her wonderfully titled talk, A Spoonful of Sugar, A Scoopful of Poop. So over to you.
Thank you very much. And I'm just going to see if I can create a laser pointer in case that's useful. So yeah, thank you very much for the invitation to talk this evening. And I'm going to just touch on a few of the background information that Rob has already given you. It's always good to drum in some of these basic facts and then sort of move into the clinical application that is relevant to my field in terms of the microbiome and cancer therapy. So just to remind you that the human body is an ecosystem, and we live alongside a variety of these microbes, many of which are bacteria, but also we've heard fungi and viruses. They mainly exist within the gut, but also other organs like the skin, the lungs, the urogenital tract, and the oral cavity. And there are a lot of these cells. I'm not going to give the answers too much. You can look at the data on this slide, but really a healthy host microorganism balance is needed for optimal body functioning. I think you're hopefully getting that message from the previous talk. So again, the gut microbiome is really important in multiple functions: the digestion, metabolism, vitamin biosynthesis, pathogen resistance, neurocognitive development, and especially our immune system and its development. Now, it is continually evolving, and in fact, if you think about it, literally just as a baby is born and passes through the birth canal, it is exposed to microbes for the first time. And you can actually compare it. There's a difference between the microbiome of a baby that's born via cesarean section and natural birth. And it's thought that this may be implicated in susceptibility to disease later on in life. Now, there are many other influences of the microbiome, and some of those we've already touched on. So ethnicity, where you live in the world, your environment, what you eat and how much you weigh, whether you take antibiotics or probiotics, whether you exercise, how stressed you are, and what other disease states you might have at the time. But equally, changes in that microbiome or dysbiosis is in itself associated and implicated with human disease causation. And some of these are summarized here. So common conditions like hypertension, allergy conditions, food allergies, eczema, asthma, inflammatory conditions like arthritis and inflammatory bowel disease, and indeed malignancy itself. So if you think about it and where the gut microbiome is sitting, it may be not surprising that a lot of work has been done to look and see whether the microbiome is linked to promoting colorectal cancer. And indeed, within the mucous membranes, the mucous lining of the gut, exist numerous bacteria, and a particular organism called Fusobacterium appears to be associated with a variety of cellular functions, which can generate chronic inflammation and abnormal cell proliferation. And abnormal uncontrolled cell proliferation is basically the hallmark of cancer. So this could be an example of a bad bug. Now, people have looked in tumours, and here this is a microscopic slide of a pancreatic cancer, and you could see amongst the blue tumour cells these little orange dots, and these are actually bacteria, a bug called gamma proteobacteria. And people don't really know necessarily how these bugs are getting there and what they're doing there, but they clearly do exist, and you can see them on an immunofluorescence picture here. The little red dots again are the bacteria. And it does appear, at least in pancreatic cancer, that these tumours with a lot of these gamma proteobacteria appear to be more resistant to conventional chemotherapy that we use to treat pancreatic cancer with a drug called gemcitabine. And there's quite a lot of work that is going on to try and understand how bacteria are interacting with response to chemotherapy. And you can imagine that if we could understand this, we might be able to target and manipulate the bacteria to improve response to chemotherapy. Now, chemotherapy is one of three key pillars of cancer treatment that have been around for a very long time. Surgery, radiotherapy are the other two. But in the last decade, a fourth pillar of treatment has now expanded our repertoire and is being used more and more to treat cancer patients. And this is immunotherapy. And I'm going to talk really about the role of immunotherapy for the rest of my time. So the modern immunotherapy with drugs called immune checkpoint inhibitors really started with some clinical trials in patients with metastatic melanoma. So melanoma is a skin cancer, and about 16,000 people in the UK will be diagnosed with a primary melanoma each year. And most people will have that removed by surgery. They might have some drugs after their surgery, but 80% of people will be cured. But unfortunately, about 20% of people may have recurrent disease, and this disease can recur in lymph nodes, in the lungs, in the liver, and in the brain. And unfortunately, this disease, if it metastasizes, historically has been an incurable condition with life expectancy under 1 year. So a really devastating disease until the advent of modern immunotherapy. And this has transformed how we manage these patients now. So I need to explain to you the basis of these drugs that we are now using in the clinic. And they're all based around this cell called the T cell. So the T cells are white cells in our body that fight infection. And activated T cells generate inflammation. And T cells have the ability to distinguish self from non-self. So if you look over here in this picture here, you can see the body organs within this human being. And T cells circulating in the body leave those organs alone, we hope, because they are self. But here we have a virus, a coronavirus, that has entered the body. This is non-self, and these are other infective organisms. And the T cells hopefully will come along, be activated, and destroy the non-self in order to get rid of that viral infection. And you and I experience that on a daily basis throughout our lives. If you get a viral sore throat, you can get swelling in the glands in your neck. And those glands are full of T cells that are activated and busy destroying the virus. And once the virus has been cleared, the glands settle down, the T cells are switched off, and everything goes back to the status quo. So we have this constant activating and turning off of the T cells that's going on in our body all the time to get rid of abnormal material. So ideally, what we'd like is for those T cells to do the same thing to cancer cells and get rid of those cancer cells if they occur. But unfortunately, they are derived from normal cells. So you can see here a bit of epithelium with an abnormal tumor beginning to grow in it. And it's very difficult for those T cells to detect this because it's too much like self. So cancers also can evolve mechanisms to suppress or deviate from immune function. So this is a real challenge for us. So here's a T cell, and the T cell sits in the microenvironment and communicates with the microenvironment via a variety of receptors on its surface. And there are a bunch of activating receptors, and there are a bunch of inhibitory receptors, keeping that T cell under control. Now, a great guy here called Jim Allison, who won a Nobel Prize a few years ago for his work in this area, worked out that you could potentially block this inhibitory receptor with an antibody. And by so doing, you could actually stimulate that T cell, put it into overdrive, and make it really go out and switch on inflammation, take off the brakes of the immune system, and go out and find and kill cancer cells. So that was the concept. And a drug called ipilimumab was developed and tested in patients with metastatic melanoma, and indeed for the first time was able to demonstrate overall survival benefit in this patient group. And some fabulous papers were published in the New England Journal of Medicine, which is a very prestigious journal from our perspective. And so here is a survival curve just to demonstrate to you where at time 0, we take a bunch of patients and we give them a treatment. And here these are metastatic melanoma patients. And if they get conventional chemotherapy, unfortunately, over 2 or 3 years, most of them will die. But when you give them ipilimumab, the immune checkpoint inhibitor, yes, again, the majority of people die of their disease. But here, the survival curve plateaus out. And it's this plateau that was so exciting at the time. So 1 in 5 patients were actually continuing to remain well despite their disease. The disease was being removed. They were in a remission and maybe even are being cured. So it turns out that actually another receptor on the T cell, the programmed death 1 receptor, is a much better target for immune checkpoint inhibition. And this receptor links with ligand, which is expressed by both the tumor cells and cells called dendritic cells, which present abnormal material to the T cell. And this double-pronged effect may be why this is a better target. And now there are multiple PD-1 and PD-L1 antibodies that have been developed by a variety of companies that are now being used to treat increasing numbers of cancer patients. And just to go back to a survival curve of metastatic melanoma patients, this is from one of the most important clinical trials performed to date, called the Checkmate 067 trial, where metastatic melanoma patients were given either ipilimumab, the drug targeting the CTLA-4 receptor, nivolumab, which targets the PD-1 receptor, or the combination. And 5 years out from starting treatment, you can see the proportion of patients who are still alive were 26%, 44%, and 52%. Now, from our perspective in this particular disease, this is a remarkable achievement, but you can see that there's still about half of the patients who are not benefiting from treatment. And furthermore, the other challenge that we have in the clinic is the toxicity of these drugs. So they generate inflammation in a very arbitrary way. And some of those toxicities and that inflammation can be really harmful to patients. So you can get colitis, so inflammation in the bowel, you can get pneumonitis and breathlessness, you can get thyroiditis and burnout of your thyroid gland, you can get an itis of pretty much any body system. And these can be life-changing, they can also be life-threatening. So as clinicians, we have a conundrum where we would really like to be able to predict who's going to benefit from treatment and who's going to run into problems with toxicity. And this is what we call biomarkers, and this is what we'd really like to have and we're interested in discovering. So it turns out that the gut microbiome could well be one of these predictors of both response and toxicity to immunotherapy. And over on this side of the slide, you can see a really nice summary of some work that was published a few years ago, where the stool was taken from patients who had either responded or not responded to anti-PD-1 antibodies. And the stool was transferred to tumor-bearing mice, who themselves were then subjected to PD-1 blockade. And the tumors from the mice that had received the stool from a responding patient regressed, but the tumors from the mice who'd received the stool from a non-responding patient seemed to grow. So that's a very nice observation. But what we really are interested in doing is finding out which are the good and which are the bad bugs at that detailed level. Now, there are several groups around the world who've already published in this area, and there are three key groups that I've just summarized their publications here. And two are in the USA, one's in France. And they have identified a variety of different bacteria that could be suggested to be the biomarkers of response to immunotherapy. But the problem is that there's not consistency between the data that they have generated. And that's a problem because we don't know how to use that information clinically. And some of the problems with their work may be that they've studied relatively small groups of patients, and that their analytical techniques may not be sufficiently sensitive to get really down to the heart of the matter. So you've heard from Rob that there are various phyla that make up most of the human microbiome, and the main groups of bacteria, the protobacteria, Bacteroidetes, Actinobacteria, and Firmicutes. But you can break these guys down into much more detail, and you can get down to species level. But each of these species has multiple subspecies or strains, and that's what we're really interested in finding out about. And Rob, again, has already mentioned this scientist, Trevor Lawley. And Trevor is a group leader at the Wellcome Sanger Institute, and he also now has a spin-out company called Microbiotica. And Trevor's technique for analysing stool is certainly more detailed than those groups that I've just mentioned who have already published in this area. And as Rob alluded to, what he's interested in doing is he takes the stool and he does do this culturing, and he identifies individual colonies before undertaking the whole genome sequencing. And this additional step of combining the culturing with the sequencing adds sensitivity and specificity. And we believe that this may hold the key to identifying the bacterial signatures we're after. So Trevor and I have been working together for about 5 years or so, and I have a research study called MelResist, where we recruit patients who are receiving standard immunotherapy drugs, and we know whether they've responded or not to treatment. And during their treatment, we collect various samples, and they very kindly have collected their stool. They pop it in their freezer, and then we collect it from them, and it gets sent to the Sanger Institute and now to Microbiotica for Trevor's team to analyze. And this is a very simple summary of the microbiome of a number of different individuals. Each of these little blue dots represents a normal control. This is either a PhD student or somebody working in Trevor's lab. And you can see that they're all clustered over here. The green dots represent metastatic melanoma patients who have responded to immunotherapy. And the pink dots represent metastatic melanoma patients who have not responded. And you can see that they cluster quite discreetly, which gives us hope that we can actually separate out individuals according to their microbiome. And what Trevor's team have done is they've actually gone down to that strain level and been able to identify 9 lead bacteria that appear to be implicated in conferring response to immunotherapy in our patients. And then what they've done is they've actually obtained the data from those key published studies that I mentioned before, and applied this bacterial consortium, and again been able to relatively cleanly differentiate responders and non-responders. And this is the first time that there has been that consistency across a number of different patient series. Furthermore, if you actually look in a tumor in a mouse model where a tumor is being grown within a mouse, you can see the black lines represent a tumor growing over 3 weeks. And if you expose that mouse to an anti-PD-1 antibody, then you actually get suppression of that tumor. Here, just by simply introducing that lead bacterial consortium into the gut of those mice, there has again been significant suppression of that tumor growth, which suggests that we could potentially even be using this consortium for therapeutic gain. So, the MITRE study. So MITRE stands for Microbiome Immunotherapy Toxicity and Response Evaluation. And Trevor and I and some colleagues worked hard to earn a programme grant from Cancer Research UK to run this study. And what we're really trying to do is validate the exploratory work that I've just described to you in a larger scale cohort of patients across not just melanoma, but other types of cancer as well. So we're recruiting patients who have metastatic melanoma, lung cancer, or kidney cancer. And these patients all have traditionally had quite poor outcomes in the past, but now with immunotherapy, a proportion of patients do very well. So we're recruiting a series of cohorts, 50 patients in each of these 6 cohorts. And when we have these patients all recruited, we will then analyze the data to see whether that bacterial signature is indeed predictive of outcome. And just to demonstrate what we're doing in the clinic, basically these patients come through to us in clinic where we are expecting to offer them standard immunotherapy, and we're also inviting them to consent to take part in this trial. And by consenting, which is entirely voluntary, we ask them to provide us with a little bit of stool on several occasions during their treatment. We're also collecting some blood and their clinical data. We're also collecting samples if they run into significant toxicity. And we also invite them to bring along a household member, maybe a spouse, to provide a sample as well. So we have a bunch of what we call household controls. So this study is now setting up across a dozen or more centres across the UK, and we're beginning to recruit our patients and really looking forward to getting some results in the next year or two. So just to summarise what we're really trying to do here, most of what we do in cancer treatment is quite blunderbuss. We basically treat everybody the same, and that's not the best way. We know that we need to be trying to be more personalized and to get the right treatment to the right patient, but we need good quality biomarkers to help us achieve this. So we want to be able to predict for response or non-response and predict for toxicity. And in this way, we'll be able to hopefully avoid unnecessary exposure to ineffective and drugs that are potentially causing side effects. And they are incredibly expensive. So a year of treatment with an anti-PD-1 antibody is the equivalent of £75,000 per patient. So these are really high-cost drugs that we're using. And obviously, if we do show that this bacterial consortium has therapeutic potential, then could we convert a non-responding patient to a responder, or treat a patient who's running into severe immune-related toxicity? So just to summarize, the gut microbiome appears to be implicated in regulating many types of body function, including our immune system. I hope I've shown you and convinced you that gut dysbiosis can be associated with many diseases, including cancer and response to cancer treatment. And accumulating evidence suggests that the gut microbiome may be a biomarker of both response and toxicity associated with cancer immunotherapy. We are trying to validate these exploratory findings in a large-scale study called MITRE, and we hope that out of this we may be able to develop bacterial consortia that we can use with therapeutic intent in the future. There's a lot of people behind this work at Microbiotica. I've already mentioned Trevor Lawley, but also Matt Robinson and Neville and Kevin Vervier. At Addenbrooke's and at the University of Cambridge, I work with a number of people, Grant Stewart and Sarah Welsh lead for renal cancer, Gary Doherty for lung cancer, Roy, Christine, Kate and Brent are our other clinical colleagues. David Bruce has been a fabulous study coordinator, getting MITRE up and running, and Katie Dalchow is our project manager. Nikos is our statistician. Doreen Milne and Catherine Booth did some fabulous work actually working out the patient pathway and how to collect the stool samples and make that easy for people. And Emily Barker did a lot of our initial mel-resist pilot work. And Dave Adams is another group leader at the Wellcome Sanger Institute, who's been really good in facilitating all this and brought Trevor and myself together in the early stages. And clearly we must thank our patients and their families who volunteer to take part in these kind of studies. So I'm going to stop there and hand over to Anna again, and happy to take any questions.
Thank you so much, Pippa. That was really, really fascinating. And who'd have known that such a sort of non-invasive biomarker could tell us so much? We spend so much time looking for all of the answers in the things we already have. So it's amazing to see that. And I guess amazing to have the students optimizing that protocol to make it nice and easy for your patients too.
Absolutely. It's been great fun trying to work out how to do this. We really didn't know just how willing patients would be to collect stool. It's not something that the British take to very easily. Whereas,
I can imagine.
Yeah, but yeah, we've got a good system going, and it certainly makes people chuckle.
Yeah, that's great. Fab. Well, we're getting a little bit close on time, so we'll have a few questions from the audience, and we've had some great questions coming in and lots of audience members also chatting among themselves and answering each other's questions. So one we have from Claire Dunn here is, can examining the microbiome be an early indicator of how to detect cancers like pancreatic cancer?
So it could be. I think, at the moment it's too early to know. I think there's lots of interesting exploratory data in linking the microbiome with cancer causation and detection and therapy, as I've alluded to. There's no hard and fast data to suggest that is the case, but it certainly is something that people are exploring, definitely. So, they're difficult experiments to do. But absolutely, it's this kind of thing that we do need to be exploring. I'm sure it's very trendy at the moment, and I'm sure there'll be more data coming out in the coming years.
Definitely. I'm sure there are lots of groups trying to work hard at that.
Yeah.
That's great. And we have some more questions coming in. So there's one from Trevor here. So are we looking to a future where patients' microbes can be manipulated so as to minimize drug side effects?
Yeah, absolutely. This is exactly the kind of thing that we can do. If we can find out with confidence, I think that's the key thing, is the confidence of knowing what is a good microbiome and what is a bad microbiome, and are there specific bacteria or species or strains that are really the nub of the matter, then absolutely that's the kind of thing that we would like to be able to do. And there is some lovely work that's already been done with fecal microbial transfer in people that are getting really nasty bowel problems with conditions like C. diff infections, and using fecal microbial transfer to treat that has actually been successfully done. So absolutely, these things are the way of the future.
So that line in your intro, the spoonful of poop, is a bit more literal than we perhaps thought it was.
Absolutely. No, I think that, one hopes that that there will be a nub of truth in that.
That's fantastic. Well, thank you so much again, Pippa. And there are loads of great questions. Everyone was really engaged throughout your talk, and lots of answers coming up in there as well. So we'll bring Rob back on at this point as well to have both of you here with us. Welcome back, Rob. And at this point, it would be great if we could answer the multiple choice questions that we had at the beginning. So a lot of people going for the highest numbers on all of these answers, which I think was a pretty good option to go for. So for the first question we had here, the answer is 2 kilograms. So this is a pretty hefty amount of weight when you compare it to the size of a human, or the weight of the average human. I don't know if that's something that surprised both of you when you came into this field of research, or if it's more shocking to us who aren't working so much with it. But it's a pretty massive amount.
Yeah, I guess initially, yeah, when you look at that totality next to your body weight, it does raise an eyebrow. But the more you understand where these microbes are, and the fact that of how many different surfaces of the body they occupy, or environments in the body, then it becomes not so far-fetched.
Yeah, and it's definitely, they're punching above their weight really as the amount that we have in the body, and exactly as you said, all the things they're doing for us. So if only we had more. So the next question we had was about the number of microbes in the body, and I'll pass over to Pippa to reveal the answer to this one.
Yeah, I guess it won't necessarily be a huge surprise to people that the answer is C. It's 100 trillion. It's a really big number, and that goes alongside what we've just been talking about in terms of the weight as well. So it is really quite eye-opening when you think about the fact that we really are an ecosystem living alongside these organisms. In a symbiotic way, one hopes.
Absolutely. And that leads, I think, perfectly into our final question, which is whether we should consider the microbiome a part of us. So you obviously mentioned in your talk, Pippa, about self and non-self and how the body reacts to that. But it would be great to know what you both think, whether your answer would be yes, which is our overwhelming answer from the audience, or if you'd still be a little unsure. So it would be good to know what you both think. Maybe Pippa, you could start us off.
Yeah, I think generally, I guess what we're trying to get over today is a yes, and that we are having that symbiotic relationship with the microbiome, and I think for people to understand that is a really good thing. There is a balance to be had. There are clearly potentially bad bugs, and there is clearly some issues around that diversity that Rob was mentioning. And so we do need to look after our microbiome.
Yeah, and would you agree with that, Rob?
Yes, I mean we're I would echo Pippa's comment about there is still lots to learn about the microbiome, but we are sort of beginning to consider this as yet another organ of the body, the microbiome, and we do need to look after it. We also need to think smart ways of how we manipulate it as well, through foodstuffs or very targeted addition of microbes or removal of microbes with things like phages as well. So this notion of phage therapy, I think these are interesting areas of future research.
Absolutely. Yeah. And it seems a lot of people in the audience are talking about manipulating and using this as Pippa's talked about, as a biomarker, and as you both talked about, as a way to have a healthy microbiome and therefore a healthy life. So I think we've all taken a lot away from that. So it's brilliant. But that's fantastic. So unfortunately, we don't have any more time this evening. If we asked all of the questions on here, I think we'd be on until this time tomorrow. But it's been really great to have you both. And thank you so much for really fascinating talks. So the last part just to mention this evening is the creative reactions branch of Pint of Science. So Rob and Pippa have been working with some local artists in Cambridge. And the artists that they've been working with are Loretto, John and Liza, and Kate, Jackie and Max. And all of them have been given the task of producing a piece of artwork based on the research that Rob and Pippa do and the talks that they've given this evening. So tomorrow night at 7 o'clock, back on YouTube, they are presenting their work. And you can come along and see they're really amazing pieces of arts that they've produced, and hear their interpretation of it. And the other thing that they're doing is a bit of an engagement activity that you can also produce your own pieces of art. And you can see the hashtag here, mypint21art. And if you want to share anything on any form of social media, and they may be able to show them tomorrow night at the creative reactions as well. So as well as the art that's already been done on both of your research, there may be a few more coming in from the audience as well. Which will be fantastic. So we hope to see you all again tomorrow evening at 7. So finally, just a huge thank you to both of you, and also at this point to bring on our behind the scenes team. So we have Annie Howard, who's been our master of comments this evening, has been putting all of your comments onto the screen. We have Emily Tabbitt, who's our producer, and she's been making sure everything's been running smoothly and getting everybody on and off. And Aina Piroj, who has been our Cambridge coordinator this year. So without all of these guys, this absolutely wouldn't have been possible. And without Pippa and Rob for your amazing talks that we've definitely all learned so much from. So a huge thank you to all of you. And we hopefully will see you all at 7 o'clock tomorrow to see the art. And hopefully next year we'll all be in the pub to have a cheers together at that point.
That's good.
But thank you all so much.
Thanks so much. Thank you.
Thank you.
Thank you.
Thank you.
Bye.